younger people, especially the younger people who have serious COVID-related complications, there's a high frequency of obesity in that group, and we think that one of the thoughts is that the uric acid may be playing a role in why they're at increased risk.
Hello, everyone. I'm Dr. David Perlmutter. What a special program we're going to have today. It's episode 2 of our interview with Dr. Richard Johnson. Dr. Johnson, as you recall from our last time together, is a professor at the University of Colorado doing amazing work in nephrology, the study of the kidneys, but so much more beyond that. His work looks at uric acid, the incredible deleterious effects of both fructose consumption and, as we will learn today, the body's manufacture of fructose. We actually make fructose in the body through activation of what is called the polyol pathway. That can be activated by hypoxia, by elevated sodium, by being dehydrated, by having a high glucose. And all of the downstream effects of that elevation of fructose are generally, in our modern world, very detrimental. We're going to be deep in the weeds today, I'm certain. So we'll be asking you to watch this podcast multiple times to get as much as you can out of it. His new book, Nature Wants Us to Be Fat. I've read it 3 times, and I'll probably read it again. I had the opportunity to write the foreword to his book, which was a really distinct honor. So I'm very, very excited to say hello to Dr. Richard Johnson. Well, Dr. Johnson, welcome back to the program.
Thank you very much. It's a delight to be back.
We have to be so formal. I want to again remind our viewers that this is going to be part 2. So, for them to understand the whole story of fructose from a survival perspective, our primate ancestors, our Paleolithic ancestors, etc., the uricase mutation, we covered all of that in part 1. We didn't think it was part 1 at the time, but it is, and that's we'll do this on the fly. I want to jump in. We're going to. All roads lead to Rome, so it doesn't really matter where we start, but I've been reading your book, as I told you now, for the 3rd time. I can't get enough of it. And what I'm first taken by is, most of the research that's quoted in your book is stuff that you participated in, which is unbelievable. I mean, participating with people around the world, looking at so many different aspects of this story, not the least of which was the study I pulled last night about how you demonstrated in white blood cells an incredible increase, 70-fold increase in mitochondria in people going on a low sodium, low fructose diet. That was pretty astounding.
Yeah, that was a pretty big surprise. We did not think we were going to see that much improvement in the mitochondria. But it just goes to show that, while exercise is a major way to improve mitochondria, we can also improve it by diet, and just by reducing sugar and salt, we can have an impact on our mitochondria. So that's quite good.
So that is an entree. I have 6 pages of notes here. I didn't know where we would get started, but I happen to have been reading the actual study last night. And, as is the case, you see a bunch of different names. At the very end, there's always R. Johnson there. And, it's amazing that you participate in so many different types of research. You get to the mitochondrial part, back to how fructose metabolism threatens mitochondrial function. That's going to be something I think our viewers are going to be somewhat in touch with. We've been talking for years about Alzheimer's kind of representing an acquired mitochondriopathy, and that much of what you and I have been talking about happening in the liver with respect to the metabolism of fructose, converting ATP down to AMP, etc. But this is also happening in the brain, isn't it?
Yeah, it is. So what's really interesting is when you eat fructose, only a small amount of fructose gets to the brain. So you would think that the brain would be safe from the effects of fructose to some extent. But what happens is the brain can actually make fructose. And when you eat sugar, we don't totally know the mechanism, but it seems that stimulates fructose production in the brain. So sugar and salt and high glycemic carbs all seem to drive fructose production in the brain. So it's not just the fructose you eat, it's the fructose that you make. And what's scary is that the effects of fructose in the brain appears to drive a lot of the same processes that we see in people who are developing dementia. So it causes inflammation and insulin resistance in the brain and mitochondrial problems. And what's been really a striking discovery in the last decade, as you know, has been that it isn't just the amyloid plaques and the tau protein that drives Alzheimer's, that there's a preceding phase, and that preceding phase is associated with insulin resistance. It's associated with dysfunction of the energy-producing mitochondria. It's associated with neuroinflammation. And what's striking is that fructose metabolism in the brain does all three things. And the risk factors for Alzheimer's correlate with the risk factors for driving fructose up in the brain. And what I mean by that is, like in diabetics, fructose production is turned on because high glucose levels stimulate fructose production in the brain. And likewise, high salt diets, which have been linked with Alzheimer's, high salt also increases the salt concentration in the blood. That turns on fructose production, and that presumably is going on in the brain. And sugar. And in fact, all the things that you've written about, David, the grain brain and all the effects of diet to affect the mentation and cognition seems to be linked through this pathway. So,
interestingly,
I hope you and I.
You had one literature citation in the book that was from, I think, June of 2016, Scientific Reports, where researchers demonstrated, I think it was a handful, 18 post-mortem, 9 with and 9 without Alzheimer's, that the level of fructose and its immediate precursor from glucose, sorbitol, were increased 4 to 6 fold in the Alzheimer's brain in comparison to the non-Alzheimer's afflicted brain, and also surprisingly a much higher level of glucose in certain brain areas. And it speaks to the fact that it's the utilization, it's this bioenergetic defect that we've been talking about now for the past decade or so, that seems to be so fundamental, and the downstream effects therefrom, including inflammation and upregulated free radical production or oxidative stress, compromising mitochondrial function, and then that leading to this caspase enzyme activation and therefore neuronal apoptosis, or the death of brain cells. And I often quote an interesting study that looks at being predictive by doing these FDG, these fluorodeoxyglucose studies that show brain glucose utilization being compromised in these Alzheimer's signature regions long before people are having these cognitive manifestations.
Yeah, absolutely. We think that there's an actual biologic pathway that was originally beneficial that went awry. And we've talked about this a little bit before, but fructose appears to have a role in trying to stimulate foraging. And when you feed animals fructose, they will start to forage. They'll look for food, they get hungry, they search, and they have to go into areas where it could be dangerous. And so the fructose actually works on the brain to try to decrease will power, to block deliberative thinking, deliberation, and to kind of stimulate rapid responses and to be able to venture into areas where it might be dangerous. As part of that, there's fructose probably looks like it has effects on the brain has certain areas that are insulin dependent, and the brain has some areas that are not. So much of the brain does not require insulin for glucose uptake, but there's certain regions that do. And one of them is the hippocampus, the hypothalamus, and all these kinds of areas that are important, like for memory and things like this. And if you're going to forage, you don't necessarily want to remember all the bad things about moving in, foraging in a certain area. And so, so what fructose does is it blocks the effects of insulin to take up glucose in the hippocampus, and that kind of dampens the memory and allows the animal to feel that it can go into a dangerous area, even that it may know. And so it helps the foraging response. So what happens is when we eat sugar and we make fructose, it's helping us forage, but it's doing it by decreasing the glucose uptake in these critical areas. And acutely, that's okay because it makes you be able to forage. But chronically, it results in basically an insulin resistance to this area where the cells are not getting enough fuel. There's mitochondrial dysfunction, a shift to glycolysis, which is a shift away from the mitochondria, the development of inflammation. And so if you do this chronically, you end up knocking out your hippocampus and you start losing your memory, and you start beginning to develop early dementia. And then chronically, we think it goes on to stimulate this inflammatory response that leads to the amyloid proteins and the tau proteins and the plaques. And so this is a hypothesis, of course, but it's really supported, as you pointed out, by the fact that fructose is elevated in patients with dementia. And also, the enzyme pathways like this, there's an enzyme that's turned on when fructose is metabolized. And we think it's the key enzyme that drives this survival switch, this energy shift, and it's called AMP deaminase. But that enzyme is elevated. AMP deaminase is high in the brains of patients with Alzheimer's, and so that fits with this whole story again. That diseases like that that fructose metabolism in the brain could be playing a role in causing dementia.
Well, again, it was a survival mechanism then. It allowed, during times of food scarcity, to be more impulsive, engaging in higher levels of risk-taking, because it basically disconnected the prefrontal cortex from the more impulsive amygdala-based behavior activity. We wrote a book about that called Brainwash. We talked about disconnection syndrome, and basically the adult leaves the room and you become impulsive, and I guess in the context of foraging, that would have been a survival mechanism. But why then, what is the upside then of reverting to glycolysis in the brain as opposed to a Krebs cycle production of ATP?
Well, I think it's just it's in this particular, so in most of the brain was not, it is not affected by the fructose, the insulin resistance. So most of the brain is still working with glucose uptake and so forth, because when you become insulin resistant, there's a lot of glucose that goes up in the blood, and some of the brain is using that to help, with cerebral function. So it's really about the impulsiveness and the memory that's kind of knocked down initially. And when there's decreased glucose uptake, it's as part of that process, there's also sort of a shift away from mitochondrial function to try to decrease, I don't fully know exactly why, but it's sort of like a protective mechanism to preserve ATP, to kind of reduce the amount of ATP that's needed to drive the whole process.
Is this part of the overall, I hate to say plan, but the overall mechanism then to just decrease metabolism in general as a way of preserving fuel?
I think so. That's what we've been thinking. But I have to admit, this whole area of what's driving dementia is a relatively new area for our research group, and so we're still trying to learn it. But we do recognize that the process that's driving dementia has a lot of parallels with how fructose metabolism works. And so, I think we still are trying to understand the actual, reasoning, but of why this biology is occurring. But we think that it's related to a foraging response that was initially thought to be a beneficial survival pathway, but when you keep driving it, it backfires into a really bad situation. And
Well, there was a interesting study that was in, I think, 2016 or 2018, 1,600 Japanese that were followed for 12 years. They had neurocognitive exams every 2 years, and many times they had MRI scans. And they found that dementia in general risk in the highest group of uric acid was increased 80%, and Alzheimer's per se increased 55%, and mixed dementia / vascular was increased 166% in those with the highest uric acid. That relates to our conversation, since the downstream metabolite of this fructose then is going to be uric acid. So it really seems to me that, as part of an Alzheimer's prevention scheme, that uric acid would be something we would want to pay attention to.
Yeah, and there's been a controversy about uric acid in dementia, because many patients, when they present with dementia, their uric acid levels tend to be lower. But that's because we believe that what happens is that when you start developing dementia, you start not eating as well. And there's actually a lot of people who start losing weight, and they actually start losing weight even before they often are diagnosed. And because uric acid is driven in part by food intake and nutrition, if you are not eating as well, your uric acid levels will tend to fall. So obesity is a risk factor for dementia, but it's when people present with dementia, they're often eating less, and they're often losing weight. Their uric acid levels are falling. I think in your book, you quote this beautiful study from the University of Alabama by Dr. Singh, where you point out that taking drugs that lower uric acid, like allopurinol, actually were found to protect against Alzheimer's. So having a low uric acid protects against Alzheimer's. And you actually pointed out that the paper showed that the dose matters. The higher the dose of allopurinol, which lowers uric acid, was associated with more protection than a lower dose.
And in that study, they actually also looked, I think, at febuxostat as well.
Yeah, that's right. And so exactly. That kind of study shows that, it's uric acid. It seems to be driving dementia. Lowering it seems to protect. But when you present with dementia, you may already have dropped your uric acid because you're not eating as well.
Yeah, and recognizing then that, we talk about purines being an inroad to raising uric acid as well, but really 2/3 of those purines are not dietary. They're endogenous from recycling of tissue. Presumably, if you didn't have enough bulk tissue to as a resource for purines, that would contribute to a lower level of uric acid. Let's talk about Parkinson's for just a moment, because for many years, it was noted that Parkinson's patients seem to have a lower uric acid level than others. And I have to admit that for a couple of years, I was giving my Parkinson's patients inosine to raise their uric acid levels. Now we see, I guess about 6 months ago in JAMA, that a study came out doing that. In fact, giving inosine, raising uric acid levels, and then evaluating Parkinson's patients on what's called the Unified Parkinson's Disease Rating Scale, UPDRS, and actually found it was ineffective. So I think that what you're saying is true, that there are other issues that we have to think about that may be at play here that may explain higher versus lower levels. We are confronted by that notion of uric acid as an intracellular versus extracellular antioxidant, acting as an antioxidant, versus intracellularly. How do we resolve that moving forward?
Yeah, so when you put uric acid on cells and it goes into the cell, it stimulates oxidative stress, and it's been shown in just about every cell type. So when it's inside the cell, it causes oxidative stress, and that is important in how it works. It's actually, how it drives its biologic effects, and a lot of its relationship with metabolic syndrome and increasing fat and things like that appears to be driven by this intracellular effects of uric acid. When the uric acid's outside the cell, it's also often bad because it can precipitate to form crystals, which are extremely inflammatory and can be a very significant problem. Patients with gout have these crystals in their joints, but they also we're now discovering that they can get crystals in their blood vessels and in their kidneys and other sites.
Prostate.
But, yeah, the soluble uric acid outside the cell does react with oxidants, so it can act as an antioxidant. So everyone goes, oh my God, uric acid can be good because it can act as an antioxidant. Now, we actually did a series of studies looking at this, and uric acid does block some forms of oxidative stress, but it also can generate oxidative stress even extracellularly, because when it reacts with certain radicals, it produces new radicals. And this is a problem of oxidants and antioxidants, and some of them they can become a radical themselves when they bind an oxidant. So they remove peroxynitrite, but they produce a peroxynitrite-like radical, at least transiently. So that's one problem. But they do remove some oxidants, and I think it's fair to say that outside the cell, soluble uric acid may be an antioxidant, and may function as one. There are some studies in patients with stroke where it's been shown that if you have a high uric acid, you're at an increased risk for stroke. That's for sure. But if you have a stroke, there's a group in, I believe the guy is in Spain, but he has done some studies where they've infused uric acid acutely in stroke, and they've actually think that it might provide a little bit of benefit acutely. That's probably because of the antioxidant effects, because they're giving it, intravenously, so it's extracellular, acting as an antioxidant. And it may be also engaging some of the survival pathways that might be important in this kind of setting. But in general, yeah.
Let me go back to the pro-oxidative effects, though, intracellularly.
Yeah.
And in your book, you really indicate that might have had some really good positive survival aspects related to it, being inducing oxidative damage to the mitochondria. And the discussion in your book was also at the time when you were talking about how humans don't make vitamin C, that there is seemingly, and that when fruit is ripe and we have the most fructose, the vitamin C level is at the lowest level. What is this survival advantage then to having a pro-oxidant activity of uric acid intracellularly?
Well, what happens is when you create oxidative stress inside the cell, and especially in the mitochondria, the place where ATP is made, what the oxidative stress does is it works on the Krebs cycle to inhibit it, and it inhibits a specific enzyme called aconitase. And this enzyme is involved in the ATP production that's produced through the Krebs cycle. When it does that, it shifts, it causes a stimulation of citrate, which actually activates a pathway that leads to fat production. And so what happens is that oxidative stress is actually important in the stimulation of fat synthesis. So it stimulates the cell to produce fat. It also is involved in the insulin resistance that develops, and it also blocks the burning of fatty acids, what we call the beta fatty acid cycle. So, and that's all driven by that oxidative stress. So it causes a little bit of fat accumulation, it stimulates fat production, and it makes you insulin resistant. And this is a survival benefit if you are thinking that you're going to be in trouble, if there's not enough food around, or if you're worried that you're going to, you're like an animal that's preparing for hibernation or preparing for winter. They want to put on fat stores so that they can burn that fat while they're hibernating. And so they eat a lot of berries and sugar, and that triggers this oxidative stress in their mitochondria, and that helps them take the calories that they were getting and shift them towards fat instead of towards energy. So instead of producing the ATP, we've blocked that by blocking that Krebs cycle and the beta fatty oxidation. So you're making less ATP, and instead that ATP or that energy is being turned into fat, which is a form of stored energy. So it's moving it from a usable energy that you could use immediately and shifting it to a stored energy. And that's what you want to do if you're going to be hibernating. If you are worried that you have a food shortage coming up, you want to store fat. And what fructose does is it's the nutrient that tries to tell the body that there's a food shortage coming and that you need to store fat. And so it tends to move the energy from producing ATP to producing stored energy. So when you eat glucose, you're trying to make active energy right away. But when you eat fructose, you're trying to produce stored energy like fat and glycogen. It shifts it. And the way that shift occurs is by causing oxidative stress with mitochondria that reduces the ATP produced and shifts that energy into fat and into glycogen and these other sources. So
this metabolism of fructose consumes ATP, reduces ATP down to, loses 2 phosphates down to AMP ultimately. And it's that loss of phosphate that you have indicated seems to trigger the lack of activation of AMP kinase and this alternate pathway, this AMP deaminase, that really sets the stage then for this lipogenesis, this change in metabolism, and really a lot of the downstream effects that kind of are underlying much of our metabolic dismay.
Yeah. So initially when the fructose is first being metabolized, it burns a lot of ATP. So the ATP levels start to fall. And then when that intracellular phosphate level falls, then it stimulates the sweeping away of the AMP, which is normally regenerated to ATP. So it consumes the ATP and then it prevents it from being regenerated because the AMP is swept away. And it also blocks an enzyme that helps produce the ATP called AMP activated protein kinase. So it blocks the regeneration of ATP. And then it produces this uric acid, and then the uric acid goes in and really finishes it off by causing this oxidative stress to the mitochondria that keeps the ATP production down and shifts the energy towards fat. And then what happens is when the ATP levels are shifted down, that's actually a stimulation for hunger because the body wants to have a higher ATP level. And so you end up eating more, which then provides more fuel that is shifted to produce fat. But eventually you make enough ATP that you're done. The process comes to a halt. But it's after that you've eaten more and you've stored more fat. So it's this brilliant way for nature to say, okay, normally you like to stay at this weight. But when I trigger this, I'm going to increase your weight. This is going to cause weight gain. And
nature
fat
wants us to be fat.
Yeah. Yes.
Who knew?
Who knew?
Who knew? There it is.
Yeah, there. Oh, thank
Nature.
you.
I have two. Actually, this was the original, the galley, and then you just sent. You may not know this, but I got the hard copy as well. One of the
Yeah, look.
fascinating things that you talk about. Oh, there you go. Good.
Yeah, and
Um,
thank you for writing the foreword.
I'm. It was beyond an honor. I want to talk about alcohol because there were some incredible insights that you had. The first was that in the laboratory animal during that would crave alcohol, metabolize alcohol, ultimately develop alcoholic fatty liver disease, that by blocking the metabolism of fructose, though they were exposed to alcohol, they would not get alcohol related fatty liver disease, juxtaposed upon the notion that fructose leads to this non alcoholic fatty liver disease. The point I'm making is that fructose seems to be involved in both alcoholic fatty
Yeah.
liver disease and the non NAF LD that we see so commonly these days.
Yeah, this is a big discovery. The full paper hasn't been published yet, but what Rob Lustig, who's a friend of ours, pointed out some time ago that sugar or fructose or sugar is alcohol without the buzz, and people who eat a lot of sugar can get fatty liver, and actually we were the first ones to report that association in humans. But sugar and high fructose corn syrup and soft drinks are a major risk factor for fatty liver, non-alcoholic fatty liver disease. And over time, that fatty liver disease can degenerate to cirrhosis and cause liver failure. And some people who are develop this non-alcoholic fatty liver disease can have to go on to get a liver transplant to replace their liver. It can be that severe. And it's actually one of the major causes of liver failure today is this non-alcoholic fatty liver disease. But it's been known for a very long time that if you actually look in the livers and look at the pathology, that it's very, very similar to what you see with alcoholic liver disease. And when you drink alcohol, you also get fatty liver, and over time there's some inflammation and scarring, and it can progress to cirrhosis. And the two have some of the same biochemical problems, like AMP kinase is low in both, oxidative stress is high in both, uric acid is high in both, and so forth. And so it's been known that the two are very much alike. And Rob Lustig has pointed this out, that sugar and alcohol have a lot in common. Now, it's also been known that people who drink alcohol tend to like sugar, and people who like sugar tend to like alcohol. And the two actually are very, very linked. Even like if your father liked alcohol, you are more likely to like sugar. I mean, it's really even a genetic thing. And so that the two go hand in hand. And it's also known that when you give alcohol and sugar together, that the sugar or fructose makes the alcohol more powerful. It accelerates alcohol metabolism, and the two work together. And like if we give an animal alcohol and we give it sugar, it will increase its alcohol intake. And not only that, they will get much more dramatic liver disease. So this was kind of already known. And then the blockbuster discovery, which our group did, but another group did it at the same time, was to discover that alcohol, when you drink alcohol, the alcohol activates an enzyme to make fructose in the body. It's not the alcohol becoming fructose. The alcohol is alcohol. But the alcohol affects an enzyme that's normally not active, and it gets turned on by alcohol, and this enzyme converts glucose to fructose, and you start making fructose. And it's the same problem that high glycemic carbs do. When you drink or when you eat bread and your glucose levels go up, some of that glucose can get converted to fructose. So it's another way the body can make fructose is to drink alcohol. And so it was this group from Kentucky actually did a beautiful study in humans and showed that this enzyme is turned on in the liver of people drinking alcohol, and that there's fructose in the liver of people drinking alcohol. And we had also discovered that if we give alcohol to animals, that their livers start to make fructose. Well, fructose causes fatty liver through uric acid. And when we blocked fructose metabolism, we could block the fatty liver that was induced by alcohol. So turns out that the way the fatty liver is occurring from alcohol is not from the alcohol itself, it's from the alcohol turning on fructose. And so alcoholic liver disease is actually a fructose disorder. So this was a dramatic observation. There was a group in China that blocked the enzyme that converts glucose to fructose, and they could block alcohol liver disease that way in animals too. Our paper has not yet been published yet, but it is under review and revision, and but it's a very exciting story.
So aldose reductase or fructokinase, either way, the downstream metabolism of either the production of fructose or its downstream metabolism is going to keep this fatty liver disease from happening.
Right.
Wow.
So I hope that one day we can, help people with alcoholism by trying to interrupt these pathways, and some of the compounds that you've talked about, like luteolin and so forth, which lowers uric acid, they have some effects on this pathway too. There's one called Osthol that is another supplement that we found can actually block some of these effects. We haven't tested it in humans though, and if you are listening and you want to take Osthol, read about it and follow the guidelines for what people are recommending, and don't overdose yourself on anything that has not been tested well in humans yet.
So in a very primitive way, back in the grain brain, early grain brain days, we were arguing against the consumption of refined carbohydrates and certainly simple sugar because of its effect on glucose and then secondarily insulin and ultimately insulin resistance, et cetera. But I think what you're defining here is that elevation of glucose through the law of mass action or other activation of this polyol pathway and ultimately the production then of fructose might be far more influential and might really act as kind of a unifying theory of this neurodegenerative bioenergetic failure of the brain, in that it ultimately induces mitochondrial dysfunction.
I think so. I think that the aldose reductase pathway, which is, when the body makes fructose, it has to use this special enzyme. And there's only one way the body makes fructose, only one. And it's through this enzyme called aldose reductase, or what I call the polyol pathway. So you can either eat fructose or you can make fructose, or you can do both. And the aldose reductase pathway gets turned on under conditions of stress. So it gets turned on, for example, like if you have a heart attack, the blood flow, when the blood flows low to the heart, that will stimulate local production of fructose in the heart. If you are in a situation where the oxygen levels are low and you are struggling, you'll try to make fructose. So these are like thoughts where, it's being produced initially as a survival tool. But if you turn it on in a big time way where the production's very high, it backfires because it can lead to cell death and trouble down the road. It can lead to obesity and insulin and diabetes and all these things. So overactivation of this pathway is probably an under recognized risk factor. And one of the things that drives that pathway, David, is uric acid. When uric acid goes up, it activates aldose reductase. So uric acid, in fact, we have thought that, although uric acid causes this oxidative stress and all these things that are bad, we have considered the possibility that this feedback loop to stimulate aldose reductase may be one of the stronger mechanisms by which uric acid is causing its problems. And so uric acid has all these effects that basically drive inflammation and drive mitochondrial dysfunction and all these things, but it also activates this process. It's like a feedback loop that leads to more fructose in the body. So uric acid is really a major target. I congratulate you on your book, Drop Acid. I really think that it's a wonderful book that really goes deeply into this uric acid question and ways to reduce uric acid by diet and supplements. I do compliment you. It's really a great
Thank
book.
you. I mean, for me, metabolic health has always been very important in as it relates to the brain, and never has gotten the attention I think that it needs. And then learning about uric acid from you really gave me a very, very powerful new tool in the toolbox. I see it as the lieutenant carrying out the mission. It gets the signal from fructose and other inputs, and it's then uric acid that's doing the dirty work. And really, I think that controlling uric acid is going to be really key, and as is even regulating the metabolism of fructose. But getting back to the production of fructose, you indicated that hypoxia is one powerful activator of aldose reductase, leading to the conversion of glucose into fructose, and then all the downstream issues like the weight gain. And I think people will say, well, gee, I'm not climbing K2, I'm not going to get hypoxic. And you think of the number of people who have sleep apnea, for example, whose saturations go down at night for whatever reason, and that they're gonna chronically, as you say, overdo it, be activating this pathway. That does what? That increases their lipogenesis, makes them fatter, which plays negatively as it relates to sleep apnea, making that problem worse. So it becomes a very vicious cycle, and that's one of the unique things about fructose metabolism is this whole notion of these feed-forward cycles. Unlike glucose metabolism with respect to ATP utilization, that check on ATP utilization doesn't happen with fructose metabolism, and it's amplified once uric acid, the lieutenant, says we need to keep doing this because this is a survival mechanism. Yeah. So you've talked about hypoxia. Let's talk about sitting down, watching the playoffs, and eating a bag of salted pretzels. What is that going to do to me?
It's not going to do you well. But if I may just go back to your point about the hypoxia, there are two things that I want to just mention, if that's okay, and then we can go to the salted pretzels. But
totally fine.
So I think your point about obstructive sleep apnea is so true, because during the night they get hypoxic, it disrupts their sleep. You wrote this beautiful section of your book goes on the importance of getting good sleep. Then you get this ischemia, you activate the aldose reductase pathway, you produce uric acid. Obstructive sleep apnea is super associated with gout. It's really associated with obesity. And so the sleep apnea becomes a driver of the metabolic syndrome, a driver of poor sleep, and all these things. And it kind of amplifies everything, and it's tied to this fructose metabolism pathway, to this uric acid pathway. It's all part of the same story. And so you made that point, but I just really wanted to tell you that I totally agree with you on this.
Yeah, and diabetes and Alzheimer's, obesity, all strongly related to sleep apnea.
And the other place where this really is developing a strong insight is in cancer. So it turns out that uric acid is being recognized in obesity is being recognized as being associated with cancers too. And although we think of metabolic syndrome as the main association of uric acid and of obesity, people with obesity have an increased risk for breast cancer and pancreatic cancer and colon cancer. And it's been a mystery why that is. And it turns out that when it may not be causing the cancer, but there's more and more data that the uric acid may help drive the cancer growth. And the way it drives cancer growth is as follows, what we think, and that is it's through this hypoxic mechanism. So what happens is a cancer cell, when it metastasizes, when it spreads to another organ, it initially gets there, it doesn't have a blood supply. And so it actually is kind of living in a low oxygen setting until it grows enough to actually get adequate blood vessels, and it will stimulate its own blood vessels to form to feed it. But for quite a bit of time, these cancer cells are living in a low oxygen condition. And it was known for a while that this occurs, and they call it the Warburg effect, because what happens is these cancer cells have to live on glycolytic metabolism primarily. A lot of it is through glycolysis, where they're making their ATP through a mechanism that doesn't require oxygen. And when you make ATP through the mitochondria, you need oxygen, but when you make it through glycolysis, you don't. And so to survive, they have to live in this kind of Warburg effect where they're living more on glycolysis. So guess what fuel is the best fuel for driving a shift from mitochondria to glycolysis? It's fructose. And so some beautiful work by Lewis Cantley and others have shown that that high fructose corn syrup and fructose are fuels for cancer cells. And a lot of cancer cells prefer fructose over other fuels. So liver cancer and colon cancer. And in fact, there's some papers that suggest that just restricting sugar and restricting fructose might be a treatment, might be a supplemental treatment for treating like colon cancer, metastases to the liver and things like that. And so we actually have done some studies on this. We just published a paper where the uricase mutation, remember that uricase, that we lack uricase, so we have higher uric acids. So we took mice and we knocked out their uricase, or we inhibited it, both ways. And when we give them a cancer that metastasizes much, much faster in the setting of a high uric acid and an absence of uricase. So the uric acid goes up, so they kind of become humanized. And now the cancer can spread easier. And we think it's because of this pathway where uric acid is reducing the mitochondrial function, kind of stimulating glycolysis and setting up an environment where the cancer cell likes. And so,
well, that said,
yeah,
I reviewed a study last night that made it very clear that SARS-CoV-2 virus replicates much more efficiently in a glycolytic environment.
There you go.
And when we look at that juxtaposed upon the studies that are demonstrating that risk for bad outcome, be it ventilator, ICU, or death, is certainly at least twofold increased, if not threefold, or the composite score, having all three, is threefold increase in people who enter the hospital with a high uric acid level, favoring this glycolytic environment, allowing replication. And then truly with that and this oxidative damage, one would expect kind of especially in immune cells, a far more robust activation of cytokine production.
Yeah.
So
absolutely,
it all comes.
People with obesity and metabolic syndrome are at higher risk for COVID complications. And when I'm an active physician too, and I'm on the wards. Actually, I was on the wards this morning, and the younger people, especially the younger people who have serious COVID-related complications, there's a high frequency of obesity in that group. And we think that one of the thoughts is that the uric acid may be playing a role in why there's at increased risk. And we also found that a high uric acid was associated with increased risk for, developing kidney failure from COVID, going on the ventilator, and even dying. So there's some thought that targeting uric acid might provide benefit to the COVID patient. It needs to be tested. I know groups that are thinking of running clinical trials to determine that.
Well, I mean, allopurinol, obviously, but why not just quercetin? I mean, why not,
Yeah, why
if you're
not?
still inhibiting xanthine oxidase, why not
Yeah,
go with something easier? But, I guess in this day and age, it would be, a drug study. That's who's going to sponsor it and take it from there. But anyway,
that's
I'm
right.
really anxious about my salted pretzels.
Yeah, salted pretzels taste fantastic. When I was young, I used to eat salted pretzels and beer with my friends.
Snyder's.
All yeah,
Snyderman's? What was it? Snyder's?
yeah. No, I just I have to tell you that um, I've had to change my diet significantly the more I study this whole field because a lot of things that I loved are not really good for you. And the trouble with salted pretzels is you got the pretzel, which is high glycemic, so that
Right.
raises your glucose. And when that raises your glucose, as you well know, that's not good. When your glucose levels start going up over 120, you start making fructose from it, and you're also stimulating insulin. And but the fructose we think is really not a good thing. So the pretzel has high glycemic carbs, so that's one hit. The second problem is the salt. And everyone, salt has been a very controversial topic in the literature. For a long time, it was thought to be terrible, and then some people are actually, arguing that salt can be good. And the truth is, salt can be good or bad depending on how much you're eating and so forth. But if you're eating a lot of salt, it it actually increases the salt concentration in your blood. And when the salt concentration goes up in your blood, it makes you feel like you don't have enough water. And that's why when you eat salt, you will become thirsty because your brain senses that you're dehydrated. And that the way it determines if you're hydrated or not is it looks at the salt concentration in your blood. And so if you lose water, like if you get dehydrated because you have diarrhea or you're vomiting and you lose water, the salt concentration goes up because you've lost water. So the salt goes up because the water goes down. But if you eat salt, the salt concentration goes up. Even though you may not be losing water, it will create the same signal to the brain, telling the brain that you're thirsty and that you're dehydrated. So eating salt is sort of a way of creating dehydration. And unfortunately, dehydration stimulates fructose production. And the reason is because when you produce fat, you're actually, it's a way of storing water. This was, something I really didn't know that much about. I think I had read it years ago that when you burn fat, you're producing not only energy, but you produce water as part of the when you burn fat, it breaks down eventually to CO2 and water. So you're making water when you burn fat. But I kind of just ignored that. I didn't think it was so important. But actually, animals will use fat as a source of water when they're in trouble. And the classic example is the whale. So the whale lives in a salty ocean environment, and they don't drink seawater. Whales do not drink seawater. So how do they get their water? You know how they get their water? Well, they get a lot of their water from the food they eat. So when they eat crustaceans, they get the water out of the crustacean and so forth. But they also get water from their fat. So they are the fattest animals in the world. The whales are huge in fat. And when they burn their fat, they're making water. And it may be 1/3 or so of the water they get comes from their fat, maybe sometimes more. And same thing with animals in the desert. So, you don't want to have a lot of fat if you live in the desert because it's going to make you hot because it's like a form of insulation. So the animals put the fat in their tails. So, they have these rodents with the fat is in their tail, or in the camel, they put it in a hump over their back but they use that fat as a source of water. So when you eat that salted pretzel, the salt concentration goes up.
I don't do that, by the way. I don't have people get the wrong idea.
No, I know you don't, man.
Do I want to do it? You bet. When I'm in New York and they have those soft pretzels on the side of the road that are
I love those.
Covered with salt. I used to always give in to that, but no longer.
Me too. Thank God. Yeah. No, it's so true. They really are delicious. There's no doubt about it. Anyway, that salted pretzel, when it goes up in the blood, the salt concentration, it triggers this fructose production. And we used to think of salt as just driving blood pressure. And it doesn't drive blood pressure up in everybody. But people who are older, people who have high blood pressure, they eat salt and their blood pressure, it seems to make their blood pressure worse. So all of us, do modest salt restriction for people with hypertension. And we know that people are eating like 10, 12 grams of salt a day. And so in general, there's some data that high of salt can be associated with heart disease. And so we try to tell people not to eat huge amounts of salt. But the problem is that in the last number of years, they started recognizing that salt intake also correlates with the risk for obesity. And there's all these studies now showing that if you're on a high salt intake, that it increases your risk for diabetes and obesity, often years out. It isn't an immediate thing, but it's sort of interesting. If you put a person on a high salt diet, even you can induce insulin resistance fairly quickly, like even within a week. But anyway, so there's an association of salt intake with obesity. People who are overweight or obese tend to drink less water and to eat more salt. And, I was talking to some of my friends who are hypertension specialists, and they say it's well known that that if you measure the salt in the urine of a patient with obesity, it's going to be high. And, when you're in balance, what you eat comes out in the urine. So you can kind of tell how much a person is eating by how much they excrete. And it's been kind of well known in the high blood pressure field that people who are overweight are often eating large amounts of salt. But now the data is really clear that people with obesity tend to be dehydrated. They're not water drinkers. And so what happens is it's another mechanism to drive obesity. So there's sugar and, purines and, but another mechanism to drive obesity is to be mildly dehydrated, not severely dehydrated, but mildly dehydrated. And the easiest way to do that is to eat salty food. And so salt, when we put animals on salt, they didn't gain weight for 2 months, 3 months. You put them on sugar, they're getting fat by 2 months of age. By 3 months, they're really fat. We put them on salt, there's not much going on for the first 2 or 3 months. And then suddenly, a little bit later on, they start getting fat, and then they get really fat, and they become enormously fat and diabetic. And we can show that it was due to this production of fructose, because if we block their ability to metabolize fructose, even though they're not getting any in their diet, we can block obesity. And that's because they're producing fructose in their body. So when we saw this, we realized, yeah.
No, I'm just saying there are quite a few mechanisms in that could explain the relationship of salt to blood pressure, including for example, the production of fructose through the polyol pathway, the metabolism to uric acid's inhibition of nitric oxide, leading to hypertension. You mentioned before low-grade inflammation within the kidney, even some autoimmunity involving heat shock protein. That there are plenty of mechanisms that are there to really keep the blood pressure up and keep us alive.
Yeah, and one of the big discoveries, and this was done with my friend Tom Jensen and Miguel and Aspa, one of the big discoveries is that there's a hormone that's turned on that actually can drive, seems to be important in how fructose works. And that hormone's vasopressin. And vasopressin, is a hormone that holds on to water. It's turned on in dehydration, and its main biologic action is to concentrate the urine. So it helps preserve water in the animal by reducing the amount of urine that you make. So it makes the urine pungent and yellow and odorous, and it's because it's concentrating the urine. But vasopressin works in the kidney through one receptor, but there's another receptor that vasopressin binds to. It's called the V1b receptor, and no one really knew what the function of that receptor was. And when we were giving sugar to animals as well as salt to animals, we found that vasopressin levels went up in the blood. And we actually know that people who are overweight have high vasopressin levels in their blood. And it's the test that's usually measured is called copeptin, but it's a stable measurement of vasopressin, and it's high in people with obesity. And people with obesity have elevated vasopressin in their blood, and the vasopressin is blocking or is binding to this V1b receptor, and that is important in how obesity occurs, because when you block that receptor, you can block sugar effects. And it's involved in the uric acid pathway and the oxidative stress. And it seems to be like part of a feedback loop that stimulates the fructose metabolism in the liver. But we don't have it fully understood how it works. But what we now know is that fructose and uric acid are two major players. But the third guy is vasopressin. So fructose is the nutrient, uric acid is the metabolite, and vasopressin is the hormone. And those are the three guys that are driving this whole process. And we can try to block each one. And the uric acid one is the one that we can measure so easily. Everybody should be able to measure their uric acid levels. And the copeptin might be another one that we might want to measure someday.
There you go, right?
Yeah, there you go, your uric acid measurement.
That was yesterday, 4.7.
That's good.
No big deal.
That's beautiful number.
Richard, I'm hearing a lot about you lately. I did a podcast just before our time together, and they were all over Richard Johnson and his work. It was Alzheimer's prevention or something like that.
Oh, wow.
And they were mentioning you. But I want to again thank you for your time. We could go on. I have 6 pages of notes. I've only done 1 page so far. I'm not kidding you. They're all laid out here. And gosh, I don't know where we would take it. But we're going to have episode 3, I think, sooner or later. But meanwhile, you're getting ready to launch this amazing, wonderful book, which I'm just so captivated to read. And I know that it's a prelude, though. I know that there's so much more to come that you're going to unravel. And, I can't wait till the volume 2 of that comes out, because I know you're right on the precipice of really putting all these pieces together, and it is really all coming together. I mean, I think back, as I mentioned, to the grain brain days of thinking just the glucose, but now the glucose through activation of the polyol pathway, elevated sodium, hypoxia, big belly, all that stuff feeding into ultimately this acquired mitochondriopathy as being the bioenergetic explanation. Really makes a lot of sense, and I'm so thankful for all these pieces of the puzzle.
Well, you've actually helped a lot in this whole puzzle, of figuring out the puzzle too, because this whole relationship of grains and of wheat and high glycemic carbs and their relationship to dementia and to all these neurobehavioral issues, you've really been a leader in this whole field. And your knowledge of the clinical side of it is really exceptional. And I do think that, combining forces, like where we bring in what we have found on the basic science side and what you have been able to see in your patients in the clinical side, is a really beautiful a way to merge our abilities. And we should think about how we can try to move the needle and help people through working together. I appreciate what you've done.
100%. And we're going to play guitar together.
Absolutely. Back to the USSR. That's my newest one.
Been away so long, I hardly knew the place. Gee, it's good to be back home.
Exactly.
All right, my
That's
friend, we'll
wonderful.
talk soon. And I'm sure we're going to talk probably the next few weeks as we get
Yeah.
closer to your book launch. But thanks for joining us today.
Yeah. Thank you too, David. Thanks.
Okay, bye for now. Wow. Yeah, it was deep. What can you say? It's really exciting stuff. And we are solving a lot of mysteries, aren't we? A lot of pieces of the puzzle are being put into that puzzle, and we're really starting to see it take shape. We're seeing evolution of ideas that we talked about many years ago now being reframed in the context of fructose and its downstream metabolite, uric acid, that tends to amplify all of this now negative aspects of this metabolism, which were once actually survival mechanisms. Again, the book Nature Wants Us to Be Fat by Dr. Richard Johnson, an amazing, amazing book. And I want to thank you all for joining us today. I am Dr. David Perlmutter, and we will be back soon. Bye for now.